So before we can understand the process of purine and pyrimidine synthesis, we need to understand how DNA is structured. This is the classical DNA structure that is seen in books. As we can see, it's composed of two strands of complementary DNA in the helical structure. Now, to understand the structure in better detail, we're going to break it down into its basic components. So in this depiction of DNA, we could see that the DNA strands are made up of monomers called nucleotides. So each of these represents a nucleotide. So if we pull the two DNA strands apart, we're going to get two single strands of DNA, each composed of nucleotide monomers. So this is the first strand and this is the second strand, and we put them together, we're going to get the double-stranded DNA helical form. You can see that we have two isolated single-stranded DNA, and then when we put them together, we're going to get the double-stranded DNA that we see in our cells. And you could also see that basically each strand is composed of monomers of nucleotides. And when you put these monomers together to form a polymer, this is what we call DNA. Take these double-stranded DNA and we twist it, we're going to get the classical helical form of DNA that we see. So we said before that DNA is a polymer that is made up of monomers called nucleotides, and this right here is the structure of nucleotides. So they're composed of three things. We have the pentose sugar and the phosphate, and we also have a nitrogenous base. Now the ribose and the pentose sugar are going to be constant, so they're found in every nucleotide. What separates different nucleotides from others is the nitrogenous base. The different nitrogenous bases. So we have 5 nitrogenous bases that are used in DNA. They are cytosine, thymine, and uracil, and these are called pyrimidines. And then we have adenine and guanine, and these are called purines. Now, when these nitrogenous bases are added to a ribose and a phosphate, we have something called a nucleotide. And we have 5 nucleotides that are used in DNA and RNA synthesis. They are cytidine, thymidine, and uridine, which are called the pyrimidine nucleotides. And then we have adenosine and guanosine, which are called the purine nucleotides. We take these nucleotide monomers and we put them together, then we're going to form a structure called DNA.